In this video, we're going to talk about the different human skeletal muscle fiber types. Fiber types are determined by first doing what's referred to as a muscle biopsy, sometimes called a percutaneous muscle biopsy. That's because it is across the skin. And here you can see the needle that would be used to do that. The first thing that is done is an incision of about a half a centimeter, a little bit longer, is made. And then the needle is pushed into the belly of the muscle, and the tissue is cut out and removed. And you can see that piece of tissue. It's probably not even as big as the eraser on a mechanical pencil. So it's not a very big piece of tissue. Here you can see an actual biopsy being done. And obviously, as you can see here, this is into the belly of the gastrocnemius. Very common sites include the gastroc, but also the vastus lateralis. And then what has to happen is typically they'll take that tissue sample and freeze it so that they then can take it back into the lab. Here you can see an unstained sample, and you can see that it's not possible to distinguish between the fiber types, but as stains of different pH are applied, you can see that now the fiber types do begin to stand out. Here is, again, a piece of tissue that's been stained. You can see the distinction between the fiber types. And what would happen is a grid would be laid down on this, and then all the fibers would be counted. And then, for instance, the type 1 fibers would be counted, and they'd be expressed as a percent of the total. And so what data would suggest is that in average sedentary people who aren't totally sedentary, we find about 45 to 55% type 1 fibers in arm and leg muscles. But even given that, we don't tend to find a sex difference, and so there doesn't seem to be a difference in male-female. And there is a very large inter-individual variation. And so in a given muscle, we might find somebody who has about close to 80% type 1 fibers, or we might find somebody who's close to 20% type 1 fibers. So a lot of individual variation that you see. This chart shows a lot of information about fiber types, so there's a lot here, and you have to learn what you see. And so what they're showing you, we have 3 different motor units. And so again, a motor unit is the alpha motor neuron plus all the muscle fibers that innervates. Here are the collateral branches of the axon going to different muscle fibers. And biomechanists use a different terminology than most other researchers use. The terminology that they use is up here at the top, and so they say the motor unit is fast fatigable, or fast fatigue resistant, or slow. Another common terminology that's used, which I do like because it gives you 2 pieces of information, is the one that you see here. FG stands for fast glycolytic. And so it tells us that these fibers will contract and relax fast, but they also rely mainly on glycolysis for energy. These fibers are fast oxidative glycolytic. Again, they contract and relax relatively fast, but they kind of split their need between aerobic energy metabolism and glycolysis. And then finally, the S type are slow oxidative, and so they contract and relax relatively slowly, and they depend primarily on aerobic energy metabolism. What you see under here is the most common terminology that's used, and it's the type terminology. And so you can see that the fast fibers are referred to as type 2. And the fastest are 2X, and again, they're the ones that rely primarily on glycolysis. And then still fast, but not quite as fast, are the type 2As. And then finally, the slow are type 1s. Now you can see that we start with finding differences in the motor units, motor neurons that control these muscle fibers. And so you look here and you can see that the type 2s have alpha 1 motor neurons and the type 1 have alpha 2 motor neurons. And you can see that there are differences in size here. The motor neurons that control the type 1 fibers are the smallest. And what that means is that the EPSPs that are created as they move around the soma to the trigger zone are getting smaller. But since the cell body is small, there's a better likelihood that when those EPSPs reach the trigger zone, they will be high enough to create an action potential. I probably should say, no, never mind. I take that back. Here we have larger motor neurons. The cell body is bigger, and so now that EPSP is getting smaller, and by the time it reaches the trigger zone, it may in fact not be big enough to reach threshold, and so we might not get an action potential. And so again, that means that the type 1 fibers have the lowest recruitment threshold, or in other words, it's easy to activate them. The 2Xs have the highest recruitment threshold, and it takes longer for them to be recruited. Another difference in the neurons is the thickness of the axon, and you can see that the 2X have the thickest axons and the type 1 have the thinnest axons. And that means that the action potential will get from the trigger zone down to the synaptic knobs faster in these type 2 fibers, 2X fibers. And so it's traveling along here faster. And it travels from the trigger zone to the synaptic knobs in the type 1 fibers more slowly because the axon is thinner. Now again, I want to remind you that all of these axons that you see here are heavily myelinated. They are myelinated, every one of them. All alpha motor neurons are myelinated. Now what I want you to be careful of is looking at this and saying that the axons of the type 1 fibers are longer, because that's not the case. Look, this is a spacer. We've cut out a chunk of the axon, and we don't know how long that chunk is. In fact, we cut out more here than we did here. And you have to remember that if we're sending a type 1 and a type 2 motor neuron axon down to the gastrocnemius, it has to travel about the same distance because it's going to be coming out of roughly the same area of the spinal cord. And so from that area of the spinal cord down to the gastroc is the same length. So don't make the mistake of saying there's a difference in length of axons. This is looking at cross-sectional area of the muscle fibers in the motor unit. And you can see that the type 2X have the biggest cross-sectional area. Type 1s have the smallest cross-sectional area, and the 2As tend to be intermediate. Now, I am going to tell you that there's an exception to this. Athletes who are endurance athletes that put in a lot of work and have been training for many months and probably more likely many years are focusing primarily on these type 1 muscle fibers, and they will hypertrophy some. They aren't using these very much, if at all, and so they will atrophy a bit. And so it could be possible to find endurance athletes who have type 1 muscle fibers that have a larger cross-sectional area than the type 2X. But that's unusual, and it's a key indicator that those people are endurance athletes. What this is showing you is what's referred to as a twitch response, and so we send one action potential down this axon to the muscle fibers. They contract and relax, and what we get is a twitch. And so you can see a twitch in the type 2X versus a twitch in the 2A versus a twitch in the type 1. And you can see that one of the differences is, again, speed of contraction from 0 up to the peak and speed of relaxation. And you can see that for the 2X, it's 100 milliseconds or less, roughly. And for the type 2A, it's roughly 150 milliseconds, a little bit longer than that. And then for the type 1, you can see that it's actually more than 200 milliseconds. And then also just notice here that contraction time is actually a little bit faster than relaxation time. And so again, contraction time is a little faster than relaxation time. Okay, the other thing that we get from the twitch is how much tension is generated by that motor unit. And so here you can see that for the 2X, in this particular case, it looks to be roughly 500 millinewtons that's being generated in terms of the tension. Here, tough to estimate, but it looks like it's maybe, I don't know, 180, 200 millinewtons. And here you can see it's not even 50 millinewtons that's being generated by this type 1 motor unit that you see here. And then finally what we see is the fatigability of these guys. And again, the 2Xs rely primarily on glycolysis and also the phosphagen system, and so they fatigue relatively quickly. The type 1s rely on aerobic energy metabolism, and so it takes much longer for them to fatigue. And then the 2As are intermediate in terms of fatigability. So again, there's a lot of information here, and you should know this chart. You should understand it, starting with the two differences in the motor neurons and working down to morphological differences in the muscle fibers and then functional differences. This chart contains much of the same information that we just looked at, but it also has a little bit of information about capillary supply to the muscle fibers and the mitochondria that we find there. And so not surprisingly, the type 1 fibers are very much aerobic, they're oxidative, and so they're going to get a nice rich supply of blood high in O2. So they have more capillaries and a higher capillary density, and they also have more mitochondria, which is what uses the oxygen. The type 2Xs tend to be low in terms of number of capillary and number of mitochondria, and you can see that the 2A are intermediate, and just how intermediate will partly depend on the type of training the person is involved in. Here we can see some histochemistry, biochemistry information. There is a difference in the myosin ATPase between the three fiber types. And remember that ATP binds to the head of the myosin. And when it does, it gets hydrolyzed by myosin ATPase. Well, the activity of the myosin ATPase in the type 2 fibers is high, while in the type 1 fibers it's low. And this is part of the reason that the contraction time is shorter in the type 2Xs and the type 2As. This is the calcium capacity has to do with the voltage-gated calcium channels and the calcium pumps that we find in the wall of the sarcoplasmic reticulum. Remember, the sarcoplasmic reticulum is phospholipid bilayer, and what we're going to find is we're going to find more voltage-gated channels here in the type 2 fibers, and also more calcium pumps to reclaim that calcium and get it out of the myofibril so that these fibers can both contract and relax faster. And then the information here on function and contractility we've actually already covered. Here, too, we've got a lot of redundant information, stuff that we've already discussed. We just looked at the density of mitochondria and capillary, and this means we're expressing the amount of mitochondria and the number of capillaries relative to the cross-sectional area of those muscle fibers. So that's how we get density. We express it relative to cross-sectional area. Another thing that's related to aerobic energy metabolism is the ability of the tissue to store oxygen. You know that the prefix myo means muscle, and myoglobin is a hemoglobin-like protein that can store oxygen in muscle tissue. And so not surprisingly, the oxidative or aerobic type 1 fibers, they're going to be high in myoglobin content. And the very non-oxidative type 2X, they're going to be very low in myoglobin. And again, the 2As can be medium. They can have an intermediate amount of myoglobin. And this is actually what makes the dark meat in a turkey dark. A lot of myoglobin and a lot of iron for the oxidative enzymes of electron transport. And the absence of all of that myoglobin and some fat and whatnot is what makes the white meat white. This is all repeat. We've talked about these functional aspects. But here you can see some information on energy stores within the muscles. And so again, the type 1 fibers are very aerobic in nature and not very anaerobic. And so they're relatively low in terms of phosphocreatine and glycogen. Whereas the type 2 fibers tend to be higher in terms of their phosphocreatine and glycogen stores. Triglycerides tend to be found in higher amounts in the type 1 fibers. Well, triglyceride can only be used aerobically, and so that makes sense. Here again, we can look at some enzyme activity. We've talked about myosin ATPase. Well, we also find higher glycolytic enzyme activity in the type 1 fibers because they are fast oxidative glycolytic and fast glycolytic. Oxidative enzyme activity can be somewhat high in the type 2As, and again, that partly depends on the training that people do, but the oxidative enzyme activity tends to be low in the type 2Xs. So that covers some of those characteristics. Here are some data that we can look at, and this looks at contraction time, again in milliseconds, so probably for the twitch. And now we're expressing the fiber type based on the myosin heavy chain that we find there. Myosin heavy chain 2 indicates that those are type 2 fibers. And now we're looking at the entire muscle. And so you can see that the soleus is mostly slow twitch. And as a result, the twitch takes a long time to occur. The triceps brachii on the back of the upper arm, on the other hand, is mostly fast twitch. And it has a relatively fast contraction time. And then vastus lateralis is a little bit intermediate. Here's some related data. This is shortening velocity now, so it's similar but slightly different. And so now when we express it as velocity in muscle length per second, you can see that the type 2Xs have the fastest velocity. And so not only are they going to be responsible for expressing high force, but they're also going to be responsible for expressing high power. Here we can see the force increase. And so now, again, on the x-axis, we have myosin heavy chain for type 2 fibers. And these, again, are the soleus fibers. Force increase relatively low, type 2 very low. And here we have the triceps. And so now the force increase is much higher. And so that should make some sense to you. And finally, we have fatigue. And now you can see the fatigue index shows the inverse. This is showing you that after fatiguing contractions or fatiguing exercise, the soleus is still able to generate about 80% or more of its initial tension. Whereas at the triceps, it's dropped off far more than that. It's dropped off to about 50% and less in most cases. Here you can see some data from a study done by some biomechanists. And so again, this would be the type 2X fibers, this would be the 2As, and those would be the type 1 fibers. We'll talk about maximum tetanic force in the next video, but this is the maximum amount of force we can get from a motor unit when we stimulate it really fast. And so here you can see that the twitch contraction time being very short is associated with a high maximum force, whereas the twitch contraction time being long, as in the slow fibers, is associated with a much lower maximum force. Again, you put those two things together, the maximum force and the twitch time, and it gives you a reflection of the power output, because we can calculate power by taking force and multiplying it by velocity. Here you can see some endurance data. And so again, the FR are the type 2As and the F are the type 2Xs. And here you can see this is being expressed as a percent of the initial force. And that's why the type 2Xs aren't higher. If we did absolute force in Newton meters, the type 2Xs would be up higher than the type 2As. But they've done it as a percent to kind of standardize it. But you can clearly see that the drop-off in force is much faster for those type 2X fibers than it is for the type 2A fibers. And so you don't get much drop-off over this 2 minutes. Here's a little bit of data looking at fiber distribution, again, in the vastus lateralis and in the deltoid, which is a little bit of an unusual muscle to do fiber typing on. So we don't have lots and lots of data on that. But this is kind of interesting. So we look at those kayakers who use that upper body and use the deltoid a tremendous amount and look at the high proportion of slow twitch fibers that we find for them. It's very high. And here you can see a comparison of males and females for different sports. And again, here you can see middle distance runners versus sprinters. And so again, it's interesting to see that the middle distance runners have a greater proportion of the type 1 fibers than the sprinters have. And conversely, this means that these guys will have a greater percentage of the type 2 fibers. So kind of interesting there. But the truth is, we really don't know how much of an influence genetics plays versus how much of an influence training plays. This is some older data. I'm not sure that it's great. it would be really nice to maybe get it updated. Here we see the percentage of type 1 fibers, and so we read from 100% down to 0% going this way. And so here are the marathoners. But again, look at the tremendous amount of intra-individual variation that we see here. Down here at the bottom would be percentage of type 2 fibers. Okay, and so let's see here if we can find, I guess what we have here are the sprinters and jumpers. But again, look, quite a bit of individual variation. Some of them less than 50% type 2 fibers, others up close to 80%. So that's really what this shows. And then this is for men. And here you can see similar data for women. I did tell you that this data is older. We probably could add a lot more to this now because we've probably done muscle biopsies on a lot more women than we had done when this was first developed. Here you can see this is 800-meter runners. Used to be back in the 60s that they thought that women, they're just too fragile. They can't run marathons. Well, we know that's bullshit now, don't we? So lots of women run marathons and run very fast. Here's some other data that you can kind of look at that is just comparing the cross-sectional area of the type 1s and type 2 fibers and for a variety of muscles. And I obviously don't expect you to memorize this stuff. But you can see that, again, in general, the type 2 fibers are bigger than the type 1 fibers. Let's look at the runners, though. Oh, look at that. The runners, that the type 1 fibers aren't quite yet bigger than the type 2s, but they're almost the same size. So that helps to reinforce what we said earlier about these people using these type 1s a lot and actually hypertrophying them and not using these guys very much and making them atrophy a bit. So there you can see that. Here you can see some differences between men and women. Probably not surprising that the men have slightly bigger fiber sizes than the women do, but that's not as true for the vastus lateralis, the leg muscle, for the type 1 fibers here. So much closer, but still bigger for men than for women. And again, obviously, the big thing that I want you to take away is that the type 2 fibers tend to have bigger cross-sectional areas than the type 1, and men tend to have bigger cross-sectional areas than women. And here is a little bit more information on individual muscles per percentage of type 1 fibers. And so here you can see upper arm, biceps, triceps. You can see it tends to be a little bit on the lower side, but can get up close to 50% in some cases. Let's see here. This one surprises me a little bit. Diaphragm never ever has to develop very much force, but it's contracting all day long. So I would expect it to be up closer to the soleus because that's a similar type of thing that the soleus does. But at any rate, the data that we have here would indicate that it's about 50-50. I don't have any idea what the number of subjects is for the data that you see here. And then this is a weird graph. I don't know why they didn't do a scattergram, but they didn't. And so what they're doing here is they're looking at percent type 1s versus max VO2. And you can see that there is some correlation here, but it's hard to see exactly what it is. Had they done a scattergram, we'd have a better indication of what kind of correlation we're looking at. But the cross-country skiers, the long-distance runners, they have the highest VO2 maxes, and they also have the highest percentage of type 1 fibers. But again, no idea who these people are. Are they elite? Are they high school competitors? I don't know. And also don't have any idea what the N is. How many of them are there? What we have been learning over about the past 20 years or so is that it appears that you very well might be able to change fiber types through the training that you do. Most people at this point would start to agree that you can make changes between type 2A and 2X. And so if you do lots and lots of endurance training, you can start to convert some of the 2Xs to 2As. Or conversely, if you do a lot of power training, you can convert some of the 2As raised to 2x's. What we still don't know is it possible to convert from type 1 to type 2 or vice versa, from type 2 to type 1? Unsure about that. There are people who are convinced that we can do it, but that's a really, really difficult study to do. It's a long-term study. Part of the problem with the studies that we have is they're taking people who have been an endurance athlete or a power athlete or whatever for probably 5 years or longer. So we don't know if it was that 5 years of training that determined the fiber types, or if it was genetics. More than likely, it's a combination of both. And again, we don't know which is more important. This is just showing you that when you do sprint bouts, not surprisingly, the muscle glycogen depletion is greater in the type 2 fibers. So that would indicate that you use type 2 fibers when you sprint. And then here's some similar data for more endurance type stuff where they're going for 2 hours. And now you can see that you're basically depleting the muscle glycogen in the type 1 fibers, and you're using up a lot in the type 2 and getting close to depletion, but it does take longer for that to occur. And so there's that data for whatever it's worth. In the next set of videos, we're going to start to look at some muscle mechanics and a couple of important relationships that we have, the force-velocity relationship and the length-tension relationship.